Hello and welcome everyone to Nanotemper Technologies' latest e-demo event, Prometheus Panta, the next frontier in protein stability characterization. I'm Dr. Leah Pandisha, a product specialist at Nanotemper, and today I will be your guide on this journey into our Prometheus Panta. I first want to thank everyone for taking the time out of your day to join us. And before we begin, I'd like to quickly walk you through a few tools to better orient you to our system and to interact with us in the platform. So on your screen, you're going to see a set of windows. You can move these windows around and customize your view. The icons at the bottom of your screen are going to correspond to and control these different windows. In particular, we have the resource list, where you can find the Prometheus Panta brochure and some relevant papers, along with our link for our next Panta event on March 30th. Another important icon is the Q&A box, which will allow you to submit any questions you may have at any time during this e-demo. We will answer these questions during our Q&A session with our special guest, Panta Product Manager, Dr. Charles Heffernan. So with that, I'd like to get started. I'm going to begin by introducing the Panta and some relevant applications in a brief presentation, and then my colleague, Dr. Daniel Turman, and I will bring you into the lab for the e-demo. Today, we will be measuring the NIST map to highlight the instrumental and analysis workflow. Monoclonal antibodies have been one of the most influential therapeutics in the past 30 years, and the NIST map is routinely used as the reference standard for therapeutic protein characterization. So as the pharmaceutical industry has grown, the need for more selective and versatile drugs has evolved, which increases the complexity of these desired therapeutic molecules. And this complexity translates to numerous different physical properties that contribute to the overall stability of a molecule, which increases the number of parameters needed to characterize that stability. This inevitably complicates the characterization and comparison process required to identify and eliminate less than ideal construction conditions in any workflow, whether it be biologics, structural biology characterization, gene therapy, or small molecule drug discovery. Because there are a lot of different properties that compromise the stability of a molecule, it's inherently difficult to select the ideal choice. Different methods and technologies define stability differently, therefore providing different answers on a molecule's overall stability. And on top of that, we need to distinguish the subtle differences of potential candidate molecules. So it's really important to obtain high-quality results that deliver clear, concise answers. And the specificity, resolution, precision, sensitivity, and accuracy of the Prometheus instruments allows you to make better, more informed decisions from your data on a single platform. So the Prometheus family includes 4 models. The NT48 and the NT-plex are used to characterize conformational and colloidal stability by measuring thermal unfolding and aggregation. And the new Prometheus Panta combines the advantages of the NT48 and the NT-plex and builds upon them through the addition of DLS optics. So Panta is the only instrument currently available that can simultaneously measure thermal unfolding, aggregation, and particle sizing through one thermal ramp, which effectively eliminates errors in having to run multiple experiments on different instrument platforms to get different stability parameters. So protein behavior can be complicated, and monitoring the conformational stability as well as the colloidal stability is imperative to getting the full story. So nano-DSF allows us to follow the unfolding process and identify transitions between the folded and the unfolded states, as well as any intermediate states as a function of temperature. The back reflection optics look at irreversible formation of large aggregates from the unfolded and the partially unfolded state, and DLS monitors the formation of reversible oligomerization from the partially unfolded state. And this complex behavior is now better understood by coupling particle sizing with thermal unfolding and turbidity measurements. Our Prometheus line uses nano-DSF or differential scanning fluorimetry to detect subtle changes in the fluorescence of tryptophan and tyrosine residues present in virtually all proteins. The fluorescence of tryptophan is strongly dependent on the protein solvent environment, and by following changes in the fluorescence at 330 and 350 nanometers, representative of the folded and the unfolded states respectively, thermal stability can be assessed label-free. This dual UV technology allows for rapid fluorescence detection, which provides an unmatched measurement speed and quality. And this yields ultra-high resolution unfolding curves, which allow for us to detect even small, subtle changes in unfolding signals. There's no secondary reporter fluorophore required, unlike conventional DSF, so protein solutions can be analyzed independent of buffer compositions, as well as over an unmatched concentration range from samples exceeding 200 mg per mil down to 5 micrograms per milliliter. And this allows for the analysis of detergent solubilized membrane proteins, as well as highly concentrated antibody or enzyme formulations. So to determine the presence of large protein aggregates in solution, we are going to use back reflection technology to probe sample turbidity. And back reflection measures the amount of light attenuated by a sample. So in the figure on the left here, we have the cross-section of two capillaries sitting on the mirrored surface of the sample tray. In a sample where no aggregation is occurring, the incident light is directed into the sample capillary, and roughly 100% of that light is back reflected to the detector. A sample where aggregation is occurring, the incident light is going to be reflected off those particles in solution, and not all of that light is reflected back to the detector, which effectively attenuates the light. And the amount of attenuated light is then plotted as a function of temperature, shown by the image on the right, and serves as a measure of the total aggregation in a sample. To probe oligomerization or small particles in solution, we're using the DLS optics in the Panta. And here, laser light is directed into the sample capillary and backscattered by particles in solution. And this backscattered light is going to fluctuate in intensity due to the Brownian motion of particles, and these fluctuations are measured and analyzed as a function of time. The raw data, or the autocorrelation function, describes the rate of these intensity fluctuations and determines the probability that a particle is found in the same location over time tau. As larger particles undergo slower Brownian motion, their autocorrelation function undergoes a slower decay, while faster, smaller moving particles are going to have an autocorrelation function that decays more rapidly. And by fitting this autocorrelation function, we can determine the diffusion coefficient, and from the Stokes-Einstein relationship, we can then calculate the hydrodynamic radius, or the particle size. So while the autocorrelation function tells you about the radius of your sample, it also gives information on the PDI, or the polydispersity index, which indicates whether there are multiple species present within the sample. And the PDI is a measure of the heterogeneity of particle sizes and can be used as a metric of sample quality. So in the figure on the top here, we have a monodispersed sample. And in the intensity distribution plot, we see a single sharp peak indicating there's one species present, and it's very monodisperse. In the figure on the bottom, we have a polydispersed sample, and in the intensity distribution plot, we have two distinct peaks indicating different particles with different sizes, and these peaks are rather broad, indicating more polydispersity of those particle species. To acquire a fluorescence sample signal in the Prometheus instruments, we are using capillaries as the sample container, and each is holding 10 microliters of sample. They sit directly on a thermal element, which is used to steadily heat the samples up to the desired end temperature according to a specified thermal gradient. And this direct control results in more precise temperature control with the measurements, which helps to achieve high reproducibility, not only between capillaries, but also between runs. And as the capillaries are heated, the entire tray is going to move beneath the optics in the instrument, which are constantly scanning and acquiring data points to allow for simultaneous measurements of both protein unfolding, aggregation, and particle sizing, or DLS, in a single thermal ramp. So there are two optical heads in the Panta, one that holds the fluorescence and back reflection LEDs in their respective detectors, and then the second houses the DLS laser in its detector. The Panta also comes with an exchangeable sample tray, which Dan will show you in a bit, and this allows for you to load up to 48 individual capillaries, or we have a capillary chip, which has 24 capillaries pre-mounted to the chip, which will allow you to load samples simultaneously. And these chips and capillaries are exchangeable, which allows the Panta to adapt to any workflow. So while Panta can be used for a variety of applications, I'm going to briefly highlight a few for you today. So we're going to start off by looking at a formulation development where the combination of nano-DSF, back reflection, and DLS optics can be applied to many different workflows like candidate selection, molecular engineering, lot and batch comparison, etc., to assess and differentiate protein stability and behavior. So here we are looking at Herceptin diluted in 3 different formulation buffers. And before I go into the data, I want to quickly highlight what each plot shows you. So we have the nano-DSF in the top, which is going to indicate the thermal stability and will allow you to get melting temperatures. The middle plot is going to be the turbidity measurements, so looking at large particle aggregation. And then the bottom one is the DLS optics, looking at oligomerization. So in the PBS buffer, which we see shown by this navy plot, there are 2 distinct unfolding transitions which occur roughly at 71 and 82 degrees Celsius. Large amorphous aggregates are forming from the turbidity measurements, which are concurrent with that second unfolding transition. And the DLS optics are also indicating the growth of oligomers from that monomeric protein at the same temperature as well. In the sodium acetate buffer, we have in light blue, shown here, the unfolding transitions shift to lower temperatures, and we no longer have the formation of large aggregates in solution indicated here. The oligomerization occurring is very slight, and I can tell you from looking at this data more in detail, it's a marginal increase from about 5 to 6 nanometers in radius. And then with the addition of salt to the sodium acetate buffer, shown by this purple plot here, we can now differentiate 3 unfolding transitions, which all shift to lower temperatures. There is the subtle formation of amorphous aggregates, shown by the turbidity plots, which occur concurrent with the 3rd unfolding transition. And then oligomerization also occurs, but that is more occurring at that 2nd unfolding transition about here. So by coupling thermal stability, aggregation, and particle sizing, it's really critical in helping researchers unravel the conformational and colloidal stability of their molecules and deciding which formulation provides the best stability to move forward with. Additionally, the sizing information from DLS can be applied to identify formulation or candidate molecules that stay monomeric and those that degrade and form aggregates. So we're looking at an accelerated stability study with Herceptin in 2 different formulation buffers, histidine and sodium acetate, incubated at 80 degrees for 14 hours. And we can see that in the sodium acetate buffer shown by these purple plots, there's really no significant change in the hydrodynamic radius after incubation of the sample. We have a very tight, narrow peak. So the protein is very monomeric and there's no shift in that radius and it's still pretty narrow. However, in histidine buffer, not only does that hydrodynamic radius increase in size, indicating oligomerization of the sample, but we also see this peak broaden at higher temperatures, indicating a more polydispersed sample. And lastly, Panta can also be used to confirm binders from primary high throughput screening assays. So the binding of small molecules often induces structural changes to the target molecule, which shifts both the melting temperature and the size of the protein. And these subtle changes can be screened using nano-DSF and DLS to confirm binders. So here we see the crystal structure of heat shock protein 90 alone and bound to ATP. And not only does binding increase the thermal stability of the protein indicated by the nano-DSF data on the left, but binding also induces a change in the conformation from an extended state to a very compact state, which is reflected in the shift in the hydrodynamic radius shown on the bottom right. So to conclude, with the Prometheus Panta, therapeutic thermodynamics can be characterized by monitoring protein unfolding, large aggregation, and can be looked at through turbidity measurements, and particle sizing can now be assessed with the addition of the DLS optics, which are all easily obtained and analyzed using our control and stability analysis software. And now we will join my colleague, Dr. Dan Turman, in the lab for the Panta e-demo. So Dan, could you give us a quick experimental rundown on what we will be measuring today?
Yeah, absolutely, Leah. Thank you for the wonderful presentation. So today we're going to be looking at the Prometheus Panta instrument live. So have a look here. Inside the instrument now is a NIST MAB sample, National Institute of Standards and Technologies, monoclonal antibody, and that's the benchmark antibody that folks use for new technologies to understand the precision of a technology and to get a handle on how well a technology performs in things like formulation screens and concentration, a dynamic range of concentrations for an instrument. And that's something that we're doing today as well. So we're going to look at different concentrations of the NIST MAB, and that's going to give us an idea about the change in hydration radius as a function of concentration, which occurs. And we're going to look at a formulation screen so that we can get a handle on what formulations work best with the NIST MAB, highlighting in both instances how quick this instrument gives you data that's going to help you move your science forward. So great. I'm going to go forward now and just tell you a little bit about the dimensions of the instrument, the footprint, and it's a quite small instrument and a bench space that is a little less than 3 foot deep and a little less than 2 feet wide is all you need with space for a laptop beside it. Here we have a large desktop, but the instrument now comes with a laptop. You have a nice touchscreen panel display here, and I'm going to open the instrument by using the touchscreen. In addition to being able to open and close the instrument door, which it dramatically opens in front of you here, you can also do isothermal temperature incubations by adjusting the temperature here. And so what you're looking at now is the sample loading tray that Leah mentioned before that slides under the optical unit to do the measurements. What's really great about this particular instrument is that you can take the sample loading tray off. And this is the 48 samples loading tray. I've loaded 18 here of the NIST MAB. The software walks through nicely on how to load single capillaries. So we're going to allow the software to show us how to do that. In the meantime, I want to show you the interchangeable chip cartridge. So for a chip, it's really straightforward. You have a 384 well plate, and in this plate, you can have your samples laid out across the rows. And you can take a chip, which has a directional indicator on it, and just dip it into the well. And that's a single dip. You load 24 capillaries. What you can't see is the sample going into it, but I can. This nifty thing allows you to tip the 384 well plate while keeping it stable.
And that allows the solution to be drawn through the capillaries quicker.
Absolutely. Thanks, Leah. I'm going to take the chip now, place it on the instrument. And then the metal device holder that is now on the single 48 capillary holder will go directly on top of this. And so you've loaded 24 capillaries in the time it takes to load one, and you can do 24 measurements. Because we don't have that many today, I'm going to place this back on and remove the chip. But that's great. That's all there is to the chips. They are capillaries that are attached to a plastic
comb.
Yeah, comb. Thank you, Leah.
And you don't have to take anything off the stage itself, right, Dan? It's just the lid?
Yep, just the lid. That's all there is to it. Here's all that is to put it back. And the samples stay loaded into the single capillaries as well, into the single capillary tray. So I'm going to close the instrument, and that concludes all of the work in which you have to do at the instrument. This is the only thing you touch. What I'm not showing you is that I wipe the mirrored surface down a little bit with absolute ethanol. And that's the maintenance. That's what you do before your experiment. It's a very hands-off instrument. Most is done at the software. So I'm going to close now. All right, there goes our sample. So inside the instrument, the sample is in an environment where the capillaries won't evaporate for the time in which you're going to run a thermal ramp. But if you were to need to seal the capillaries, I'll just take a brief moment to show you that we have these devices and sealant paste, and you just cap over them. It's very simple. You only have to do that if you're doing a measurement over 3 hours, and typical thermal ramps are around, what, Leah, hour, 10 minutes, hour and a half, I think?
Yep, depending what your start and end temperatures are and what your heating rate ramp is. Dan, are these capillaries disposable or can you reuse them?
So they're disposable. We offer disposable capillaries in single form in a glass package like this, and it's best to throw them away. They're quite inexpensive per sample, and we can get anyone that's interested in knowing the per sample or per measurement fee, we can get that to you as well. Just let us know. All right, I'm going to now share the software with you.
So Dan, are there any other sample prep considerations that we need to keep in mind, or is it really just wiping down that mirrored surface?
In terms of being at the instrument, that's it. But there are things that need to occur downstream that you'll need to think about, and that's or upstream. You need to worry about your protein sample and the purity and cleanliness of your buffers. So you must use a filter to filter your buffers, keep them very clean, no dust. And this is all for the dynamic light scattering portion of the instrument. Clean buffers with no large particles will give you much better DLS. I will say that also when you load the capillary, be mindful of having no bubbles in the sample. You don't want to have bubbles in your capillaries, and they will be introduced if they're in your sample. So you can give your sample a quick spin before to remove bubbles, excuse me.
Right, so maybe thinking of filtering or centrifuging your samples prior to loading. And the other thing you want to be mindful of is just making sure that if you are specifically, and this is specific to DLS measurements, nano DSF, it's a bit more kind of just load and go. But since DLS is so sensitive to particles in solution, you want to make sure you cover any of your pipettes or plates that you're going to be preparing samples in.
Definitely. That's great practice to get in the habit of.
Yep.
So Leah, you can see my screen now, right?
I most certainly can.
Excellent. Those of you have a look, this is the beginning Panta control software that we'll be using today to interact with the instrument. And then the data that we collect will be used in the analysis software, which is a different package. When you first open the software, you have the diagram of the instrument here, and it's going to walk us through how to open the drawer, how to remove the sample holder, and place single capillaries. This is exactly what I did when I put the 18 capillaries in the 48 sample holder. So you load your samples, and then you place the tray back onto the samples, and you take a discovery scan. So this discovery scan is going to give us an idea about the signal at 350 and 330 that we're getting from the instrument. It only takes a few moments, and it auto detects where in the sample tray that you have samples. Things to remember for DLS: keep your sample concentrations for a small protein above around 0.4 to 0.5 milligrams per milliliter. As you get into larger proteins, things that are much bigger than, say, lysozyme, they scatter more, so you can use much less concentration. For nano DSF, you can actually go down to 5 micrograms per milliliter with our high sensitivity capillaries. So what we have here is a good look at our concentration dependence of the NIST map. here in these non-samples, and then these non-samples are going to be our formulation screen. So this is at a high pH, pH 9.2. This is at pH 7.4. This is at pH 6, and they're different buffers. So glycine, tris, and citrate. So we can already see there's a difference in the total fluorescence output. But as long as we are between 100 fluorescence counts and 20,000 fluorescence counts, we're going to get perfect data. And that's a very large range. So the dynamic range of the instrument is, in this case, an order of magnitude and concentration, but you can go much further. And then you also have the ability to look at different conditions that may dull your fluorescence. Just zoom in really quickly. In black, we have 330, and then in blue, we have 350. I just want to show you that quickly. In the software, in the bottom plot here, we have the scattering in magenta, and then that's from the DLS. Those are raw counts, and then in green, the turbidity of the measurement, and those are raw counts as well. You can select to see single capillaries or all of your capillaries. On top, toggle between chip or single capillaries. And at this point, you can run a new discovery scan if you want to. Instead of using an auto detection of the sample power and the capillaries that need to be there, you have the opportunity to do this manually. And you can also, yep, go ahead, Leah.
I was just going to say, I think the auto detect is actually really awesome, especially if you have different samples in your capillary. So if you're not running 48 of the same concentration or the same buffer, just from the example shown here, the instrument will find the perfect excitation power to span whatever you have in those capillaries.
Yeah, it finds the Goldilocks excitation power for you, definitely.
Yep.
And then DLS laser power, I always keep it 100 unless we're doing very, very large particles that are on the order of 100 nanometer or larger, in which we have to cut the signal back some. Then you can add a measurement. So now we're going to move past the initial phase of making sure we have enough protein in our capillaries to deciding whether we want to do standalone DLS or thermal ramp with DLS. So first, let's do size analysis, standalone DLS, and I want to show you some things in the software that are pretty cool. All right, first of all, we have acquisition parameters here. I'm going to toggle this on and off, and you'll see the acquisition per capillaries change and the duration change. So you have two choices. This instrument's not going to stall and get into a loop of acquisitions. You can only do 5 acquisitions per capillary over 500 milliseconds, or you can do 10 acquisitions over 5 seconds. Those are your choices. We're going to do high sensitivity. We're going to keep the temperature isothermal at 25 and DLS laser power at 100. And let's go through and annotate our samples. I hope you're watching because this is going to be quick. There you go. So NIST MAB, I've selected all of them and made a single typing move, and it's all good to go. I'm going to select our solvent now. This is going to be in our tris-based NanoTemperTech MST buffer, actually. And then these three are going to be in a citrate, a glycine buffer that I think I've put in here, or a glutamate buffer, excuse me. And these are going to be in tris.
And while Dan's filling those out, if you have, say, 48 samples and you already have them in an Excel worksheet for other assays, you can export or copy and paste that data into the chart here.
Absolutely, which makes things much easier. This is just a table format. You may ask yourself, who cares about what the buffers are? Well, it's very important because we are doing a measurement of the hydrodynamic radius of the protein, and we get that by the instrument measuring directly the diffusion coefficient. And we use the relationship, the Stokes-Einstein relationship, where the diffusion coefficient is proportional to the inverse of the hydrodynamic radius times the viscosity of the solution. Here, we automatically calculate viscosities. If you want to put different solvents in, we have this manage solvent option. That's going to let you go through and select many different types of solvents that have their own calculated viscosities already here based on concentration. We have a nice database for that. Or you can just write in, your buffer has a 1.003 for viscosity. So it's water with just a little bit of salt or something. And then you can name it. We can also add annotations. Let's say we wanted to add pH here. It's very simple. This is going to place pH as a column. And for the sake of time, I'm not going to fill that out. I just want to show you that it's really easy to do.
And these annotations come in really handy when you go to the analysis software, and if you have a lot of samples, it allows you to merge samples based on the annotations and kind of organize your data really well that way.
Absolutely. And you can also, in the analysis software, use those annotations to merge. Did you mention that, Leah? Maybe you did, but
that's good. It's something important we need to reiterate.
Yep.
So Dan, can you decouple the DLS measurements with thermal unfolding, or do you have to do, if you're doing like a thermal unfolding measurement, do you need to do nano-DSF, turbidity, and DLS?
Great question. You can uncouple them. You do not have to do the DLS measurement, but the entire set of samples will be subject to the thermal ramp. So if you're not doing DLS, you can either choose to do it before and after, or you can choose to do it during the ramp, which is the only instrument that's on the market that I know of that gives you the flexibility to have all three of those options.
Gotcha. And when you are doing the thermal unfolding, and I know we're getting ahead of ourselves a little, but while this runs, when you do the thermal unfolding measurements, those nano-DSF, turbidity, and DLS, those are all acquired simultaneously at the same time. It's not first nano-DSF, turbidity, and then DLS?
So they're acquired simultaneously, and the really nice thing is that's all attributed to the engineering of the instrument and how the optical device within the instrument interacts with the samples and takes the measurements. So there is a slight shift because of the location of the optics and the DLS measurement acquisition and the thermal nano DSF and back reflection that's reflected in the software. If you look closely, you'll see the points are offset just a little. And that's only because we know the temperature of the capillaries exquisitely close and around 0.1 degrees Celsius. So we can actually calculate exactly what that temperature shift would be.
Great, thank you.
The two optics. So let's have a look here. This measurement is just pure DLS, and we give you the size or intensity distribution or size distribution as a violin plot. So you're seeing each acquisition build on itself. And then the cumulative analysis is a single point, as it should be. It's a discrete measurement where you take and fit the ACF, which we can see here in the Acf Details. Looks beautiful. Single fit, and you get a single intensity distribution. Go ahead, Leah.
And that ACF is the autocorrelation function that I had mentioned earlier in the presentation.
Yep.
And the cumulative analysis fit that's being performed here is assuming an average hydrodynamic radius and average diffusion coefficient.
Absolutely. And then your distributions would come from a different fit of each acquisition here. This is not only each acquisition, this is also every capillary. And what you will begin to see is that at high pH, the NIST mab is quite unhappy. And that's just a point to show you that even with samples that aren't perfect, you're going to get data in which you can interpret here. So, Leah, if you don't mind, I'm going to switch to data that I've already pulled out of the oven and it's already ready to look at. So it's this exact experimental set that we've already measured.
That would be great.
Cool.
And as Dan's pulling that up, this size analysis measurement that we're doing is a high sensitivity, as he mentioned, and for 18 capillaries should take roughly 15-ish minutes. So it's still a pretty quick measurement, even with high sensitivity, doing 10 acquisitions per capillary.
Definitely. And you can see your progress here. It's actually going through and telling you what capillary it's on, what acquisition at that capillary it is. We may have to stop this in just a minute. That's okay, to move on to the thermal. But before we do that, I want to show you the results of this. And I think it's really cool. So our concentration dependence of the measurement, first of all, I just pulled up the Panta analysis software, and it automatically merged the replicates. So we did every condition in triplicate. The software automatically merged them. This is the look that you have. You can go deep into the DLS data by looking at individual acquisitions, just like we could in the control software. Or you can collapse this and look at kind of your 30,000 feet view, all of the merged replicates together. So let's look at a comparison between 7 milligrams per milliliter, 5 milligrams per milliliter, and 1 milligram per milliliter. So as you can see here, believe it or not, there's a small shift in the hydrodynamic radius just in between these particular measurements. And it turns out that the NIST MAB is exactly, or has been well characterized to be a little over 10 nanometers in its radius, in its diameter. So we get a radius around 5, and then as the concentration goes down from 10 milligrams per milliliter, which is what the measurements were done at the National Institute of Standards and Technology, as you go down in concentration, that correlates to an increase in your size of your radius. And perhaps Charles may touch on that later in the Q&A. If you would like to learn more after that too, we can talk about it. Great. Now let's look at our worst condition compared to our best condition. So here is the pH 7 milligram per milliliter in green, very tight peak, but at pH 9.2, it looks very broad. You have some states that are occurring out here, some oligomerization or even aggregation that's occurring. And then there are many states within this peak, many more that are in this one. So that's, if you were a formulations chemist, you would think deeply about why that's occurring and how you can improve your formulations. If you're a structural biologist, you could use this information about formation of perhaps a long sought after protein complex that you haven't been able to find, and now you found the condition in which it stabilizes it, etc. Great. Did I miss anything with the DLS software? Leah?
I think that was a great highlight. We can touch on some of the functionality of Panta analysis when we show some thermal unfolding data, but for the time being, that was excellent. Thank you, Dan.
Yep, thank you. I'm going to cancel this measurement. As you can see, our data is building up. The bad condition here is beginning to fall out like we've seen in the cooked data. So that's nice. So I'm going to stop this measurement and we're going to move on to thermal unfolding. So as soon as the instrument is ready, it's going to allow us to choose thermal unfolding. There we go. And for those of you that are going to stand in front of the instrument, it's nice to know that it transfers the data for you. You're not going to have to re-input those things, but you do have some more parameters to think about. So excitation power is going to be the same, but you need to think about your temperature slope in degrees per minute. Let's do that at 1 degree per minute. And your starting and ending temperatures. 25 to 95 is standard, but if you know your protein unfolds at a certain point, you want to speed up the temperature ramp, you can do that. Let's do dynamic light scattering. You can toggle this on and off. So have a look here, 1 hour and 10 minutes. In this case, toggling on and off your dynamic light scattering, it's not going to change your time, but it does change your data point density. because we have to stop and do the DLS measurements at a particular temperature. So if we're not doing that, we can do nano DSF more times. We have an indicator here that says our data point density is at a sufficient amount, which is anything greater than 1.9 points per degrees. And it's all based on your temperature slope. So let's see. You can do what looks like 7 is going to be not quite enough. 6, 4 degrees per minute, 3 or 2. So that's pretty much the only limitation you have.
And what Dan is showing here that's really nice is if you set a temperature ramp that is too high, so you're not getting enough data density, because you want a lot of data density in your transitions to get an accurate melting temperature, the software will actually flag and say, you know, hey, hang on a second, maybe rethink your parameters. You might want to acquire some data points and slow down your ramp.
And in addition, if you still have some questions about that parameter, you can click on the Nanopedia. And the Nanopedia is going to give you all the information you want about it, and probably some information you don't even know you want.
This is one of my favorite resources of the instrument, the Nanopedia. It's basically a Wikipedia for your Prometheus Panta.
Yep. One last thing here, Leah, I think I want to mention the refolding ramp. So if you want to cool the instrument in the same speed in which you raise the temperature and you want to take data, you can do that to watch for refolding in special cases. So let's start the unfolding ramp. All right. The instrument is now making some sounds where the samples are moving and the optics are preparing to do the thermal ramp. And you also get this nice indicator display here. The current temperature is 25. You have an hour and 10 minutes remaining. With that being said, let's have a look at the analysis software that we already have the data in. So the analysis software does it 2 different ways. You can do size analysis or thermal analysis, and we're going to look at thermal now. And just as before, the software is merged to replicates, and now you're looking at the ratio of 350/330, the turbidity, and the cumulant radius. Some things that I like to do, and the software allows you to do to visualize this information. Let's look at the first derivative of the ratio. I like to normalize the turbidity. It's important if you're comparing between multiple measurements. And then the cumulant radius here is going to give us some great data. So you see these hash marks. Those are measured inflection points and onset parameters. So here we're measuring 2. But let's say we really think there's something here. We can add a region of interest. And we can start it here and go to here, and it's going to calculate and fit the data. And for this particular one, it's too small to come up. But for some, it looks like it fit it here. If we don't like that particular measurement, we can remove it. We can remove the region of interest here. Or if we do like it, we can apply it throughout the whole data set. If you're interested.
Oops, sorry.
No, that's okay. Go ahead, Leah.
I was just going to say it's really great to select that region of interest, and if, again, you have 100 samples, to just automatically apply it to either the ones you want or all of them without having to do it for each sample.
Definitely. And you can get rid of the grid lines.
Dan's favorite part.
Yes, it's much cleaner, but no one believes me or agrees with me. Let's see here. Another really nice part of this software is key parameters. Let's say I'm interested only in the inflection point number 1, which is the first inflection point measured by the instrument. And I want to see how it tracks throughout all the samples. Select it, apply it, and you get a measurement of that here. This is really nice. So it looks like at 5 milligrams per milliliter, we have an inflection point at 61 degrees, and then at 1 milligram per milliliter, that inflection point is 54, etc. These are completely reasonable in terms of when a difference in inflection point will occur when you have a different concentration. So the TM changes. Let's have a look at that. Showcase how the instrument can allow you to look at multiple samples. So this is 7 milligrams, 5 milligrams, and 1 milligram. We have some problems in this particular measurement, so let's go look a little closer. So all of these warnings are saturation in the DLS detector. Looks like we have one sample that aggregated on us very early, and we're going to exclude that and look at it so we can look at it more closely. So 7, 8, and 9.
What's that shaded region?
Yeah, great question. That's the variance in the measurements. So that's going to be your difference between the individual replicates. And then the line is the mean of that. And let's look at, it's pretty busy. So I'm going to turn off the markers just for a second. And I'm going to take away our region of interest. And I just want to focus in on this particular change here. That's the change in size based on concentration. Not size, sorry, melting temperature. It's a well-known phenomenon. Let's look at our really bad sample. Oh my goodness, yeah. So this sample at high pH, NIST is not happy at all compared to this. So I would say this peak, we can turn the markers back on. This peak and this peak are the first transitions. So we destabilize that protein significantly. And it also has some just aggregation that occurs very early as well.
And Dan, I don't think you mentioned this, but there are a variety of markers here. So we have the onset of melting, the onset of aggregation or turbidity, and the onset of oligomerization for the DLS data, as well as the inflection points for each transition in the nano-DSF data as well. So that's why you're seeing a variety of markers here. And also when you overlay any data, you'll pull up the marker for each sample.
Absolutely. Thank you for mentioning that. I'm just scrolling through the data. Some of this is, like this particular sample is beautiful and the markers are really nice. You're looking at a antibody that is given a very canonical unfolding profile where the CH2 domain unfolds, Fab domain unfolds and drives the colloidal stability. And then the last thing that generally unfolds is the CH3 domain in the FC region. So that is best seen in the NIST at 1 milligram per milliliter. As you increase the concentration, those begin to be blurred. And one last thing, Leah, I think I would like to mention is the Acf details. So let's select one capillary, and it's going to tell us throughout the entire thermal ramp what the Acf detail is. So these are real DLS measurements with the same detail as standalone DLS taken during this thermal ramp. It's really incredible.
Could you scroll down a little bit? I'd like to highlight that from this we also get the PDI value,
right.
And you'll see some alerts here as well as some things values highlighted in orange, which are just quality checks and telling you to better investigate what's going on in some of these samples because there could be potential issues. So it's alerting you to do a little bit more in-depth analysis on particular samples or capillaries.
Definitely. And just to add to that, we are in this region here. So this is at 90 degrees and things are just really unfolded, aggregated, and the detector is saturated. But if we go back, we can see that the polydispersity index for this protein is 0.08. That's pretty much the average across. That's a very, very, very good. Radius is around 5.5. That radius balloons at once it begins to unfold, and the colloidal stability falls. Yep.
And Dan, last thing, could you please highlight any exportation functions on the software? since I know that's going to be a big question for a lot of people. How easy is it to export this data?
Absolutely. Excellent question. Let's quickly look at it. If you have a look here, there are export points in the software itself that's going to allow you to export images that are either in publication quality or presentation quality directly from the you see what you get here type of export. You can export all the data in the table that gives you inflection point, scattering, cumulative radius, etc. But the most powerful is the batch export. So we can do selected rows. Here, I just want to export one so it doesn't take too much time. Let's look at all the raw data. Let's get the data table. Let's get it exported as an Excel sheet. And let's get the charts. We'll just look at the current selection of charts, but you can do any other charts. It's going to be a PNG and in XLS format, XLSX format. Key parameters will be exported. So this has infinite possibilities in how you can go about doing your export. It's really quick. Asked if you would like to look at it. So let's have a look at the sample. So this is our tris buffer, our second replicate, our NIST map. And what we're seeing here is a PNG that's giving us exactly what we have there. And then this is all the data. This is a single wavelength data that you can filter out, but it's the raw data taken from the detector. Everything the instrument measures, we allow you to get from the instrument. It's one of the most transparent softwares and instruments that I've ever worked with, and I think you're all going to find out there.
Yeah. So Dan, is there anything else you wanted to highlight for our audience today?
I think that's it for me. Thank you for your attention and time to show you the Prometheus Panta. Leah, thank you for helping walking us through it and the great presentation.
Great. Thank you, Dan, for highlighting the ease of use, the software, and just the experimental workflow of the Prometheus Panta. I'd like to now introduce my colleague, Dr. Charles Heffernan, who will join us for our Q&A session. Charles is the Panta product manager here at Nanotemper. So Charles, one of our first questions is, does your instrument give the molecular weight of different oligomers?
Yeah, that's a good question. And the answer is tied up in with what the technology is and how it works. So what Prometheus Panta measures is a phenomenon known as dynamic light scattering. And a dynamic light scattering measurement is fundamentally looking at the Brownian motion or diffusion of particles. This Brownian motion or diffusion is not a property of the molecular weight or the mass of a particle or protein in solution. It is actually a property of the size of that protein or particle, as well as any sort of, let's say, the viscosity of the solution itself and any interactions between that particle or protein and the solution around it. Therefore, when you make a dynamic light scattering measurement, what you receive as your output is a measurement of that hydrodynamic radius or the size of particle that moves through the solution. This displacement or volume of the particle and its mass are, of course, closely intertwined through the density of whatever the molecule is that you are measuring, but they are not a one-to-one linear correlation. So when you make a measurement with any dynamic light scattering measurement, what you get is the size of the particle that is in solution that is being measured.
Great, thank you. Can you explain the concentration dependence in the hydrodynamic radius value, and are there any detrimental effects of doing measurements at, say, 10 mg per ml?
Yeah, this ties in really nicely with something that was being discussed earlier or being featured earlier, which was the measurement of that NIST MAB at different concentrations. And in fact, this type of measurement of measuring a molecule at different concentrations is precisely what a KD measurement or that diffusion interaction coefficient measurement is. It is looking for precisely what you're asking about, the effects of different concentrations of that protein on its diffusion coefficient or how it's moving through solution. The ways in which a protein's concentration can affect its diffusion coefficient or the way it's moving in solution is through potential attractive or repulsive interactions between molecules, those intermolecular interactions there. If you have an attractive interaction, proteins or molecules can sort of hold on to each other and slow themselves down in solution so that when you measure them, they appear bigger than they actually are. Conversely, they can also have repulsive interactions. So every time that protein A encounters a different type of protein A or a different protein A floating around in solution, they repel each other and they appear to be moving more quickly, equivalent to a smaller molecule. So when you make that dynamic light scattering measurement, they appear to shrink as you increase the concentration. And that is actually what groups look for when they are making that diffusion interaction coefficient measurement.
Why does the protein have a different melting temperature at different concentrations?
Yeah, this ties in again with another topic that was discussed previously, and this is those thermal shift assays, right? So you can think about changing the concentration of your protein as being equivalent to introducing something else into solution that might stabilize or destabilize your protein. So if your protein has, let's say, stabilizing self-interactions with itself, as you increase its concentration, it's kind of like adding a compound that might be stabilizing the protein. Conversely, if a protein destabilizes itself when in close proximity, it's kind of like adding some sort of compound that destabilizes your protein and decreases its stability or TM with increasing concentration. And that's precisely the same phenomenon that you're seeing when you change that concentration and increase it.
Thank you. What is the cost per sample for an individual capillary as well as for the chip setup?
Yeah, those costs are relatively equivalent, and it depends upon whether a group is working with our standard capillaries or the high sensitivity ones. But when working with the standard capillaries, the cost per capillary or per sample being measured is around 70 to 75 cents per sample, and that is the same for both the non-chip version as well as the chip version, which enables one to work in the high throughput format with a plate.
Could you quickly mention the difference between the high sensitivity and the standard caps for me?
Yeah, so the high sensitivity capillaries are designed to be used when measuring proteins at lower concentrations, closer to the limit of detection. So that is for groups who are really trying to either conserve sample or measure in the more extreme condition. Like, let's say you have a very potent molecule that you want to characterize at a very low concentration, or you have very limited amounts where even measuring just a couple of microliters could still compromise other parts of your characterization, and you would prefer to dilute it down to a lower concentration. Of course, groups who make those decisions have to keep in mind what we were just talking about previously with regards to the effects of concentration on both the measured hydrodynamic radius and the stability of the molecule. Those properties could both change as you change the concentration. They're not guaranteed to, but it's a possibility. So these are just considerations groups need to keep in mind when designing their experimental setup.
Thank you. Can I use DLS to measure particles that aren't spherical, like rod-sized particles?
Yeah, definitely you can. When making a dynamic light scattering measurement, really all you're looking for is that diffusion or Brownian motion of proteins and particles in solution. And all molecules will diffuse. It doesn't really matter what your shape is. You just have to keep in mind that when making a dynamic light scattering measurement, there is a mathematical assumption that that diffusion coefficient that you are extracting is from a molecule that is spherical in shape. And if you measure a molecule that is not spherical, that has, let's say, some sort of elongation or of the molecule, so it's no longer spherical, that can actually increase the apparent diffusion coefficient. So therefore, those molecules could appear to be smaller than the more spherical versions, right? But of course, when you're thinking about how this plays out when performing different types of measurements, typically what groups are looking for is when I change the, let's say, the experimental conditions, I change my buffer or my excipients that are in solution, how does that affect the stability or the polydispersity of my sample? You're comparing within that singular protein itself, and so therefore all of the results are self-consistent and still comparable. Those changes that we were mentioning with regards to the change in the apparent size due to the non-spherical nature of some proteins only comes into play when comparing across different proteins. Right?
So you're kind of looking at trends here.
Precisely.
So one of our next questions is actually referencing the resolution of the Panta. So with some specific applications looking at proteins in micelles, monomers, and dimers, and then also non-purified protein samples. So could you comment and elaborate on the resolution of the Panta?
Yeah, there's a couple of different aspects we need to keep in mind when unpacking this question, and those tie in with the fact that Prometheus Panta has different measurement modalities or optics inside of it. We have our nano-DSF optics, we have our back reflection optics, as well as our dynamic light scattering optics, all of which have slightly different answers for those questions, right? So if we start off with the dynamic light scattering optics, it is commonly said, and perhaps you might have heard, that DLS is a low resolution technique. What is meant by this is that to be able to really resolve different size particles, and what I mean by resolve is I mean to have a baseline separated peaks, right? To have baseline separated peaks so that we can see we got two different size particles in solution. You need to have a size difference between your two different types of particles of approximately 3 to 5x difference. So as you can see, when we're talking about the world of proteins and micelles, the answer can really depend on whether or not you're able to actually have this sort of baseline resolution between the two on the relative size of your proteins and your micelles. Now, one of the things that we have shown with Prometheus Panta that is really nice and powerful about the technique is even when you lack that baseline resolution between your different populations of different size or different types of particles in solution. For instance, when you are working with monomers and dimers, you are still able to, with the Prometheus Panta's DLS, identify the shift in the location of that single peak. So you can't see, let's say, necessarily what is a monomer, what is a dimer, and resolve those two different peaks. But you can see that the shift in your peak location from the monomer might shift from, like, let's say, around 5 nanometers in hydrodynamic radius for your monomer out to around 6 or 6.5 for your dimer. And this is something that can be reproducibly measured over and over again to identify that transition between the two. Of course, with that shift, you aren't able to necessarily precisely say what percentage of your sample is still in the monomeric or dimeric state, but you can still be able to discern the stoichiometry of those interactions, right? Now, when it comes to the other optical modalities, let's say the nano-DSF optics or the back reflection optics, those additional additives don't really add anything to, let's say, the background signal for the nano-DSF, right? When you have those micelles in the background, really the only signal you're going to see in your nano-DSF is from your protein of interest. So it's not going to impair your ability to characterize the effect of those micelles on your protein. And if we're talking about the world of monomers and dimers, again, with the perspective of the nano-DSF optics, you would perhaps see a change between the two if there is stabilization or destabilization when you make that transition from one state to the other, right? Much like that thermal shift assay that we were discussing previously.
Gotcha. Thank you. What is the run-to-run reproducibility of antibody TMs on this instrument?
Yeah, so I think Dan covered this pretty nicely when he made those duplicate measurements of the NIST MAB or previously, right? But going beyond duplicate measurements, we have actually shown this very robustly or extensively with an application note looking at the reproducibility of the nano-DSF and the DLS optics in a couple of different app notes, where you can see that your intra-assay reproducibility on the instrument is within 1/10 of a degree Celsius in terms of TM determination. Of course, that reproducibility does depend upon the sample itself. If you have a protein that is, let's say, not very well folded or has some variability in of itself, you might see some shifts in the TMs, but that, of course, is a function of the protein and not the instrument there. As long as your protein is highly thermodynamically stable with a narrow unfolding peak, you can achieve that plus minus a tenth of a degrees Celsius within the single instrument. When it comes to inter-instrument or inter-assay reproducibility, the instrument still performs very, very highly, as we've been able to show that you can have TMs within plus minus a third of a degree Celsius between different instruments.
Oh, that's really close. And then last question for our session is, does the mirror used for back reflection cause a problem for DLS?
Yeah, so really, to cut to the answer first, the answer is no. And that is entirely down to how we have designed the dynamic light scattering optics in Prometheus Panta to really work in combination with the entire optical system, right? Of course, you could imagine if you were to look at some sort of light scattering measurement from directly above the mirror, you could foresee a situation in which the mirror would impair your ability to determine what light is being scattered and what light is being reflected. However, we have designed the instrument to measure in such an angle where the mirror plays no role in the light that is coming back to the detector whatsoever. To do this, we are measuring at an off-angle axis where we are not directly over the mirror, and then our detector is also in a back scattering orientation, so any light that would be reflected off of the mirror goes in a completely wrong direction from where the detector is located. So this is entirely down to instrument design to be able to achieve the goals that were set out, to be able to measure all of these different properties simultaneously. That's a great question.
Thank you. So that concludes our e-demo event on the Prometheus Panta today. From everyone at Nanotemper, I want to thank the audience for tuning in today. We thoroughly appreciate the opportunity to present our instruments and technology. We will have an on-demand recording available for all of the audience members. That's going to be sent in a follow-up email. And if we didn't get to any of your questions submitted today, someone at Nanotemper will also be reaching out to you in regards to that question. I'd also like to again invite everyone to our next Panta event on March 30th. Charles and myself are going to have a discussion on some literature regarding or obtained on the Prometheus Panta. And just have a nice and lovely rest of your day.